Hi, my name is Uri Ben-David. I'm an assistant professor at Tel Aviv University, and I'm happy to give this tutorial, which will be both an introduction to the research conducted in my group and a summary of our most recent published work. In my lab at Tel Aviv University, we study cancer genetics, focusing on a relatively understudied trait of cancer, its imbalanced chromosome number, a condition known as aneuploidy. We all have two sets of chromosomes, one that we inherit from our father and one that we inherit from our mother. Chromosomes must be segregated equally into the daughter cells during cell division. And when errors are introduced into this process, this can result in an imbalanced chromosome number. Missegregation of chromosomes occurs frequently in cancer, and this was first observed over a century ago by Theodore Boveri and Marcella O'Grady-Boveri. This is what chromosome missegregation looks like when using modern microscopy, from a study by Stefano Santaguida and Angelica Amman. The outcome of chromosome missegregation is an imbalanced number of chromosomes in the cell, the phenomenon that we call aneuploidy. Aneuploidy is a hallmark of cancer. About 90% of solid tumors and 3/4 of hematologic malignancies are aneuploid. On average, over 25% of the cancer genome is affected by aneuploidy, more than is affected by any other type of genetic alteration. Aneuploidy is a fascinating phenomenon because it is mostly embryonic lethal and has detrimental consequences during development. Yet, it is a characteristic feature of cancer cells. This has become to be known as the aneuploidy paradox. I became interested in studying aneuploidy as a PhD student with Nissim Ben-Venisti at the Hebrew University. We found that stem cells tend to acquire the same aneuploidies that characterize tumors of the same tissue origin. This suggests that the fitness advantage of specific aneuploidies depends on the cellular context. We also found that the most common aneuploidy in pluripotent stem cells, trisomy of chromosome 12, can increase the proliferation and tumorigenicity of the cells. So in some contexts, the right aneuploidy can be tumor promoting. In my postdoc with Todd Golub at the Broad Institute, we found distinct patterns of aneuploidy across cancer types. Here you can see gains in red and losses in blue. In glioblastoma, for example, there are recurrent gains in chromosome 7 and losses of chromosome 10. Each tumor type has its own characteristic patterns of aneuploidy. This does not necessarily mean that these events drive cancer, but it is consistent with positive selection for specific aneuploidies during tumorigenesis in a lineage-dependent manner. Aneuploidy, however, remains an elephant in the cancer room. Despite being a hallmark of cancer, we lack mechanistic understanding of its roles in cancer initiation and progression, and we still lack therapies that directly take advantage of this fundamental difference between normal cells and cancer cells. A major reason for that is that aneuploidy is challenging to study. By definition, it encompasses multiple genes, as there are hundreds and sometimes thousands of genetic elements on each chromosome. It is also context-specific, as I've already explained, and it is hard to model. Even today, in the CRISPR era, it is far from trivial to introduce or remove chromosomes into and from cells. At the same time, aneuploidy provides a unique lens into tumor evolution, because at the cellular level, it is a discrete event. A cell is either euploid or aneuploid, unlike gene expression or methylation, for example, that are gradual. It is also unique to cancer cells, as the normal cells in our body are largely diploid. And finally, aneuploidy can be determined using various methods, from classical cytogenetics to CGH and SNP arrays, DNA, and even RNA sequencing methods. In the past few years, first as a postdoc and then as an independent investigator, I've been working on studying aneuploidy using various cancer models. And at the same time, studying the faithfulness, stability, and evolution of the cancer models themselves, using aneuploidy as the readout. In a first study, published already a few years ago, we characterized the aneuploidy landscapes of genetically engineered mouse models, GEMs, and revealed a driver-specific pattern of aneuploidy. We observed marked differences in aneuploidy patterns across mouse memory tumors initiated with distinct drivers. For example, breast cancer induced by p53 is associated with widespread aneuploidy in multiple chromosomes, whereas breast cancer induced by HER2 is associated with a specific aneuploidy. We took advantage of these recurrence patterns in order to narrow down the region of interest in one of the most recurrent chromosomal changes in human breast cancer. Here is how it goes. We found that HER2-induced memory tumors tend to lose a copy of mouse chromosome 4. Mouse chromosome 4 is synthetic to 4 human chromosomes, among which only the short arm of chromosome 1 is recurrently lost in human memory tumors of the HER2-enriched subtype. So, assuming the same gene or set of genes underlie the recurrence of this loss in both species, we could narrow down the region of interest and use gene expression patterns of our orthologs to identify and later validate a tumor suppressor gene that cooperates with HER2 during tumorigenesis. In another study, we followed aneuploidy evolution in patient-derived xenografts. These models are taken from an immune-competent human environment and transplanted into immune-deficient mice, often subcutaneously. So we suspected that this might change the selection pressures and consequently the aneuploidy landscapes of the tumors. We found that in 90% of the PDX models, very few passages were sufficient for the emergence or disappearance of aneuploidy. Interestingly, the rate of changes was similar between PDXs and freshly derived cell lines. Our study was recently challenged by the work by Wirt and colleagues who profiled the copy number of hundreds of primary tumors and their matched PDXs, and reached the conclusion that the copy number profiles are highly conserved. Wirt al. used Pearson's correlations as their main measure of similarity. Here, for example, you can see that each PDX is most similar to the primary tumor from which it was derived, as one might expect. However, we have recently reanalyzed the DNA copy number data from Wirt al., and we found that it actually corroborated our original analysis. A median of over 10% of the genome was altered between primary tumors and their matched patient-derived xenografts. And much of this difference was due to differential aneuploidies. Moreover, in cohorts that had primary tumors and PDXs from two different passages, the higher passage was significantly less similar to the primary tumor than the earlier passage, in line with model evolution. We therefore think that the difference between our conclusion and that of Wirt al. is due to the different ways in which we define similarity and dissimilarity, and the different interpretations of the results. What can we learn about aneuploidy as a driver of tumorigenesis from the fact that some aneuploidies can gradually disappear when the tumor is transferred to the mouse environment? While recurrent aneuploidies can play a driving role, they are not essential for the tumors, since subclones without them do exist. This does not mean that they don't drive tumorigenesis, as we often see subclonal driver mutations as well. It also teaches us about the importance of context. The adaptive value of aneuploidy depends on the cellular context. Unlike point mutations that mostly have a neutral adaptive value, aneuploidy mostly has a negative adaptive value, and therefore, when environmental conditions change, the likelihood that it would be selected against are high. We next turn to human cancer cell lines, the workhorse of cancer research. In a study that we published a couple of years ago, we documented the genomic evolution of cancer cell lines and showed how their diversification led to phenotypic consequences. In a follow-up study, we recently found that the evolution of the cell lines is not random, but can be determined by experimental interventions. When Cas9 is expressed in cell lines, even in the absence of a guide RNA, the p53 pathway is activated. And this promotes the selection for p53 inactivating mutations, which in turn can lead to greater aneuploidy tolerance and genetic instability in the affected cell lines. One of the byproducts of these studies is that we can identify strains of the same cell lines with and without specific recurrent aneuploidies. For example, loss of chromosome 17p is the most common aneuploidy in breast cancer. Some MCF7 strains have lost this arm, whereas others retain it. The strains that have lost it have the expected reduction in the expression of 17p residing genes. And this can serve as an isogenic-like system to interrogate the functional consequences of this aneuploidy, as we currently do in the lab. In my lab, we're currently interested in a few key questions. First, we're interested in understanding the selection pressures that shape the aneuploidy landscapes of cancers. Second, we're trying to identify genes that drive the recurrence of specific common aneuploidies. We apply experimental and computational tools to dissect genomic, functional, and clinical data from patients and from models in order to identify, prioritize, and validate drivers of common aneuploidies. The third line of research in the lab is the selective targeting of aneuploid cells. We try to tackle this challenge from two complementary perspectives. On one end, we want to identify Achilles heels of specific recurrent aneuploidies. On the other hand, we want to reveal vulnerabilities of highly aneuploid cells, regardless of the affected chromosome. The long-term goal here is to devise strategies to kill cancer cells by targeting the consequences of the abnormal number of chromosomes. An example of the first approach is coming from a study led by Stephen Corsello from Todd Golub's group. In this study, we screened thousands of FDA-approved drugs against hundreds of cell lines using a DNA barcoding approach. We observed an association between the status of the long arm of chromosome 16 and the response to the drug disulfiram. Interestingly, disulfiram is a metal chelator, and chromosome 16q harbors a cluster of metallothionein genes, which are metal-binding proteins important for metal homeostasis. We validated that knockdown of MTF1, a regulator of this cluster, sensitizes the cells to this drug. But what about the other side of the spectrum? Multiple studies have shown that a high degree of aneuploidy is associated with poor prognosis and drug resistance in cancer patients. In a recent collaboration led by Angelica Amon that was published a few days after she passed away, we demonstrated that slower cell division is one mechanism by which aneuploidy increases resistance to chemotherapies. However, work from multiple labs has shown in recent years that aneuploidy also elicits a variety of cellular stresses that are independent of the specific affected chromosomes. These include mitotic, replication, proteotoxic, and metabolic stresses. The question then is whether we can also find increased drug sensitivities that are induced by aneuploidy and would allow us to selectively eliminate aneuploid tumor cells. So we wanted to ask whether high levels of aneuploidy might make cancer cells more susceptible to some genetic or chemical perturbations. To examine that, we turn to the Broad Institute Cancer Dependency Map, which provides a unique resource to link genomic features with cellular dependencies. However, this resource did not include at the time information about the aneuploidy landscapes of the cancer cell lines. To fill this gap, we followed an approach that was previously applied to human tumors, and we assigned aneuploidy scores to about 1,000 human cancer cell lines. Here, for example, you can see a subset of breast cancer cell lines that are highly aneuploid, as is shown by the gains in red and the losses in blue, and another subset of breast cancer cell lines that are euploid. This classification allowed us to compare the cellular dependencies between the euploid and aneuploid cell lines in an attempt to identify Achilles heels of aneuploid cancer cells. Of course, when performing this analysis, we had to consider how to best define aneuploidy and account for the basal ploidy status of the cell lines. We also had to account for various confounding factors associated with aneuploidy, such as p53 status or cell lineage. But with the appropriate controls in place, we were now able to examine the genetic and chemical dependency data and identify cellular vulnerabilities of cell lines with a high degree of aneuploidy. We found increased sensitivity of aneuploid cancer cells to the genetic perturbation of MAD2 and BUB1B, two core members of the Spindle Assembly Checkpoint. This increased dependency was not confounded by cell lineage or by p53 status. Interestingly, we found that the expression of these two genes was significantly lower in the aneuploid cell lines, and the dependency was significantly correlated with the gene expression levels. These findings immediately drew our attention, since the Spindle Assembly Checkpoint, also known as the mitotic checkpoint, is the major mechanism by which cells ensure the proper segregation of chromosomes during mitosis. The seg proteins are responsible for delaying the transition from metaphase to anaphase until all chromosomes achieve bipolar orientation in the mitotic spindle. It has been known for many years that seg inhibition could lead to chromosomal instability and aneuploidy, and that this can result in tumorigenesis. But what we see here suggests that the opposite direction may be true as well. Aneuploid cells might be more dependent on seg activity than euploid cells. We then turn to compare the drug sensitivities between aneuploid and euploid cell lines. There are no chemical inhibitors of BUB1B and MAD2. However, there are multiple inhibitors of MPS1, the kinase that regulates the Spindle Assembly Checkpoint. Surprisingly, we found that aneuploid cells were more resistant to 3 different MPS1 inhibitors across 3 independent drug screens. We validated this increased resistance in our hands. This is important because MPS1 inhibitors are currently in multiple clinical trials, either as single agents or in combination with chemotherapies. However, there are currently no known biomarkers that could help stratify patients for such treatments. So we see increased sensitivity of aneuploid cells to genetic perturbation of seg core members, but increased resistance of the same cell lines to multiple MPS1 inhibitors. This could be due to the difference between genetic and chemical inhibition, due to the targeting of different proteins, or due to different time points of the assays. To solve this conundrum, we turn to an isogenic system of near-diploid and highly aneuploid human colon cancer cell lines. We found that aneuploid cells were more resistant to MPS1 inhibitors in 72-hour and 96-hour assays, recapitulating the results from the chemical screens. Importantly, we also found them to be more resistant to short-term genetic knockdown of all 3 genes, BUB1B, MAD2, and MPS1. We then turned to another isogenic system, generated in a completely different way, and found highly similar results. These findings suggest that the difference between the genetic and chemical screens is not due to the nature of the perturbation or the protein being targeted, leaving us with the hypothesis that it results from the different time points of the assays. We therefore performed longer-term experiments and found that after 2 weeks of exposure, the trend switches and aneuploid cells indeed become more sensitive to the knockdown of BUB1B, MAD2, or MPS1. So if we look at it at day 5, the aneuploid cells are more resistant, but if we look at it at day 14, they become more sensitive. We observed the same trend with the chemical inhibitors of MPS1, At day 5, the aneuploid cells are more resistant, but after prolonged exposure, they become more sensitive. These data indicate that it is indeed the time point that matters. Why is it then that the aneuploid cells seem more resistant to SAC inhibition at first, but then become more sensitive? We hypothesized that aneuploid cancer cells could overcome the SAC inhibition more readily than diploid cells, but this results in aberrant cells that exhibit severe proliferative defects. We therefore followed the karyotypic evolution of the cells upon spindle assembly checkpoint inhibition using single-cell DNA sequencing. In these plots, each row represents a single cell, and each column represents a chromosome. As expected, prior to treatment, both the karyotypic heterogeneity and the degree of aneuploidy were higher in the aneuploid cells compared to the near-diploid cells. At day 3 of SAC inhibition, all cells were chromosomally unstable, but the resultant heterogeneity was significantly higher in the aneuploid cell lines. After 2 weeks of exposure and drug removal, the surviving cells in the near-diploid populations had the same near-diploid karyotype and low karyotypic variation as that seen in the untreated cells, suggesting that the original near-diploid karyotype was more fit than the aneuploid karyotypes induced by the drug. By contrast, in the aneuploid population, the fewer surviving cells had highly aneuploid and heterogeneous karyotypes. These results suggest that the euploid cells can indeed overcome the prolonged drug treatment, whereas the aneuploid cells cannot select for a fit karyotype. So why do the aneuploid cells accumulate more chromosomal aberrations upon SAC inhibition? We found that the aneuploid cells slip more quickly from mitotic arrest upon treatment with the spindle-poisoned nucadazole, and also when treated with SAC inhibitors. However, they exhibit more mitotic aberrations, such as micronuclei formation, failed cytokinesis, and multipolar spindles. We also noticed that the spindle geometry and dynamics are altered in aneuploid cells. So we asked what might be the molecular basis of these spindle defects. By analyzing the expression of various spindle-related proteins, we found that the mRNA and protein expression levels of one particular kinesin, KIF18A, were lower in our aneuploid cells. Interestingly, depletion of KIF18A was previously shown to result in spindle geometry alterations very similar to those that we observed in our aneuploid cells. We therefore turned back to the cancer cell line data and found that the aneuploid cells were significantly more sensitive to knockdown or knockout of KIF18A. In fact, it was one of the top 10 genes most preferentially essential in aneuploid cells. We then knocked down KIF18A in our isogenic cells, and indeed, we found that the aneuploid cells were more sensitive to its depletion. Finally, we wanted to ask whether the dependencies on KIF18A and on the spindle assembly checkpoint were indeed functionally related. We thus overexpressed KIF18A in the aneuploid cells. We found that KIF18A overexpression reverts the short-term resistance of aneuploid cells to SAC inhibition, demonstrating a functional link between these two dependencies. In summary, this leads us to propose a model in which euploid cells have a normal spindle structure and functional KIF18A. When SAC is inhibited in these cells, they arrest in mitosis, resulting in relatively few mitotic aberrations, which they can eventually overcome. In aneuploid cells, in contrast, the spindle structure is abnormal, at least in part due to aberrant KIF18A function. When SAC is inhibited, the cells can quickly overcome the mitotic arrest, which makes them seem more resistant to the inhibition at first. However, these cells acquire multiple mitotic aberrations, which eventually results in their proliferation arrest and cell death. This project was a successful collaboration between 7 laboratories across 5 countries, And this came particularly useful in the past few months because we were all going into and out of lockdowns and quarantines. So at least at every time point there was a lab that somehow was still open and we were able to keep the science going. I'd also like to mention two papers from the Stumpf and the Gainem labs that reach similar conclusions to ours with regard to the vulnerability of cells with extra chromosomes to KIF18A depletion. Together, I think that the three papers really demonstrate the attractivity of KIF18A as a molecular target for anti-cancer therapy. Some of what I discussed with you today was work from my postdoc with Todd Golub and Ramin Berukim at the Broad Institute. And I would like to thank my lab, of course. This is what our group meetings look like these days. I'd like to especially highlight Yael, the first author on the Nature paper that I shared with you, and Michal, the co-first author on the PDX analysis that I discussed earlier. And I would also like to thank our funding agencies for enabling this research. Lastly, we are looking for both experimental and computational trainees. We're currently particularly interested in a computational PhD student or a postdoc for an ERC-funded position. So please feel free to contact me if you're interested. Thank you for your attention.